Hello everyone, welcome to the Biochemistry INI-CET May 2023 recall session. So analyzing the questions of INI-CET exam May 2023, what we could find is that most of the representation was from pre and paraclinical subjects. So in my subject, I'm discussing about 10 questions asked from biochemistry, so that I'll be discussing now. We'll see one by one. So before going to the questions, I just want to analyze the questions that were asked in biochemistry session for future exams, because if you are preparing for other exams, this analysis is important. So what I can find is that about 10 questions from biochemistry. Out of that, one question was a direct repeat question, a frequently asked question, that is the thermic effect of food, otherwise a specific dynamic action. So that was one direct repeat question. There was one new question asked, which was not asked in any of the previous years, neither in NEET or in INI-CET. So that was a question on Barth syndrome, which is linked to cardiolipin. All other topics, like the questions, were not direct questions, like direct repeat questions, but all the topics are previous year question asked areas only. For example, mutation detection technique, then Roth-Yaras test. Many times that question was asked, not the same question, but similar question was asked. Then ketone bodies, very important topic. Then vitamins, as you all know, it is a must learn topic for every exam. Karyotyping. So karyotyping, there was a question on the different banding techniques, which is the banding technique used to localize the centromere. So that was a question asked previously. So this was not actually a banding technique that was asked, different types of banding techniques, but the steps of karyotyping was asked here. Similar question about the steps of recombinant DNA technology, where isolation of DNA was asked one or two INI-CET back, like I think in the last 2022 INI-CET. Then RNA sequence. The sequence of DNA is given, and what is the RNA sequence? I haven't seen, like in INI-CET previous year, last 5 year, I haven't seen this question, but this is a very commonly asked question in the previous All India exams and AIMS exam. This is a very important topic that we should be learning, that is from the DNA sequence, how the RNA. Then Pompey's disease, you all know that glycogen storage disorder. I always say that's a very important topic. Then collagen. Collagen, very important every time. Either collagen disorder is asked. Last time the structure, like the synthesis of collagen, from there the post-translational modification was a similar question from the same area, like from the collagen. So that is also every time, it's a very important high yield topic. So all the topics were asked from high yield areas only. So the important message is that never miss any high yield areas of biochemistry. And as nowadays, you know that all the questions are applied questions and scenario based question. But in this INI-CET, scenario based were 3 or 4, but rest were not scenario based, but still applied aspects of biochemistry were asked. One question I say that it's not an applied aspect, but pure core biochemistry is the enzyme absent in like liver, because there is no ketone body utilization in the liver because of the absence of which enzyme. That was a topic which is asked, which is not an applied aspect, but it's a core biochemistry topic. So the take home message is that applied areas of every topic in biochemistry is important for these INI-CET exam, and never miss any previous year question topics. So that is what you have to learn whenever you are preparing for any exam. You find out which are the previous year high yield topics, concentrate on those areas, revise it as many times possible, revise it. Okay, so we shall discuss the questions. Question 1 is, which of the following method can detect alteration in DNA sequence? The options are restriction fragment length polymorphism or restriction fragment length analysis, flow cytometry, pyrosequencing, and FISH. So here the answer is a multiple answer type of question. So the correct answer here is restriction fragment length polymorphism can detect alteration in the DNA sequence, also pyrosequencing, but not flow cytometry and FISH. So the answer here is A, which is A and C. So we'll see one by one. So we will discuss what is this restriction fragment length polymorphism or restriction fragment length analysis. We know that restriction enzymes are enzymes which can cut at specific palindromic site. Okay. So whenever, that is, if there is a alteration in the DNA sequence which create or abolish a restriction site, then it will cause a difference in the length of the restriction fragments produced by the action of restriction enzyme. So in this picture we can see that this is a DNA. So in this DNA, what is shown here is a restriction enzyme. So this restriction enzyme normally has 3 restriction site. Fine. So if this restriction enzyme is acting on this particular DNA, how many fragments we will get? Yes, we will get 1 and 2. So 2 fragments we will get if this restriction enzyme acts on this particular DNA. Suppose what will happen if there is a mutation in the second restriction site which abolished that restriction site. So what will happen? This restriction enzyme can act here and here, which means instead of 2 bands, we can get only 1 band in a deceased person. Okay. So when we do a restriction enzyme digestion, and then we are doing a electrophoresis where the DNA fragments that was produced by the restriction enzymes are fragmented and that is separated using an electrophoresis. Then that specific sequence, that is, we specifically know that. Only thing is that we should be knowing what is a mutation, otherwise we cannot do RFLP. So we are using specific radioactivity or like fluorescent label on those band which we are expecting an abolishment or a creation of a restriction site. We are using that. So specifically we can see some fluorescence or a dye which can detect a color or a fluorescence, anything we can detect. So in a normal person, if the restriction enzyme is acting, how many bands we'll get? We'll get 2 bands, but in a diseased person we'll get only 1 band. So in a normal person 2 bands and in a diseased 1 band. So can a restriction fragment length polymorphism or restriction fragment length analysis detect a DNA alteration? Yes, it can detect a DNA alteration. Now the next correct option was pyrosequencing. What is this pyrosequencing? Pyrosequencing means that whenever, if this is a DNA fragment, okay, single stranded DNA, we know that these are different nucleotides, okay. So to this we are adding deoxynucleotide triphosphate. Suppose this is A. Okay, we know that the A it can join with what? AT. So it can definitely join with a DTTP. So DTTP will come here when it hybridizes with this A, when then you can see that here the T will be joining, that is the base which contain thymine is joined here, but at the same time what happens simultaneously is that what is attached is what comes is a nucleotide triphosphate, but 2-phosphate, that is when this is attached, it is attached as a DTMP, or simply we can say TMP, that is thymidine monophosphate only is attached, which means the 2-phosphate is released. So whenever a hybridization takes place, a pyrophosphate is released like this. Okay, this is a basic thing that we should be knowing in hybridization. This basic principle we are using in a technique called as pyrosequencing. How is it? That is, we have a DNA template here and various deoxynucleotide triphosphates are added. 4 types of deoxynucleotides are added. So whenever one nucleotide is incorporated, what is released? A pyrophosphate is released. So this pyrophosphate is converted to adenosine triphosphate, and this adenosine triphosphate, when acts on what you called as a dye, which is actually, it can cause light and it can produce a fluorescence. So a fluorescent dye, which is called luciferin, is converted to oxyluciferin. For that we have an enzyme called luciferase. So this enzyme, when it acts, it emits light and we can detect it. So which means that whenever if there is an adenosine here, whenever it is attached with a T, DTTP is coming, then DTMP or TMP will join and PPI is released. So PPI will be released. So that PPI will produce the light. So what will happen if there is an alteration in the DNA? If there is an alteration in the DNA, when we add the next nucleotide triphosphate, if there is suppose what we are expecting here is a G. Okay. So what we add is G always pair with C. So what we add is DCTP. Fine. Okay. So when we add DCTP, it should bind, right? But if a mutation happen here, so that it is mutated to suppose T, it is mutated to T. Now, when we add C, will it join with this T? What we expect is G, but what actually there is T. So, when we add C, it will not bind, right? So, if it is not binding, can it release pyrophosphate? No, it will not release pyrophosphate. So, will there be any light here? No, which means that if there is an alteration in the DNA sequence, and if we are adding the when we are adding the correct nucleotide triphosphate, we expect a light. But if there is no light that is produced, which means that there is an alteration in the DNA sequence. Okay. The other techniques what is given us, so that is pyrosequencing and RFL. I have explained that. Flow cytometry. What is flow cytometry? Flow cytometry is a technique used for cell counting, cell sorting, and to study the different phases of the cell cycle, like the viability of the cell, etc., can be studied. It cannot study the sequencing in the DNA or DNA sequence alteration. We cannot study using flow cytometry. So to just explain what is flow cytometry, is that first we have a sample. So the sample is containing cells are there in the sample. For example, a blood sample or any sample, cell sample. Then we are staining the cells in a suspension, or the cells that we want to study, we are staining, and it is passed through like a file like this. What you can see that it is passed. So here we can see many cells, but when this comes here, it is passing, the cells are passing one by one. Okay, so the red color is a stained cell, that is a cell that we are studying. So, when a stained cell passes through this outlet, there is a laser light source. So, this laser light source is targeted, or it is projected here, it is emitting a light here. So, when it hits the stained cell, we know that the fluorescence is emitted. It is a stained cell, fluorescence is emitted from the stained cell, and that is detected. So that we are studying we can count the cell. Also, we can we can sort the cell. The surface markers of the cells can be studied. Then we can also study like flow cytometry is a generalized term where are different techniques under the flow cytometry. So advanced techniques are there. So based on this advancement in the flow cytometry techniques, we can do all these kind of or all these are the uses. I've told you, counting of cells, sorting of cells, the cell cycle phases can be studied. Okay, so that is very shortly about the cells, the flow cytometry. So what we have seen is the cells are stained, and the reagents that are closely associated with an intended cell process. So if we are studying a cell process, we stain only those cells, and this cell, like the stained cell, will emit a reporter signal. So this helps in cell counting and cell sorting. Then fluorescence activated cell sorting, that is FACS. It is a newer flow cytometry technique which differ from conventional flow cytometry. It's the difference is that it has the ability to sort. It has the ability to sort and accurately purify the specific cell population. So it is newer advancement where sorting is also possible. Then in specifically asked in genomic studies, what is the question is about a DNA alteration. So it's a genomic study. So flow cytometry in genomics is used only in monitoring the cell cycle phases. So that is what is the use of flow cytometry. And finally, what is asked in the question is FISH, fluorescent in situ hybridization. Fluorescent in situ hybridization is the technique to study the structural abnormalities as well as numerical abnormalities in the chromosome, and it can detect deletion. It cannot detect a DNA alteration where like a DNA alteration mostly means that it is a point mutation. So one nucleotide is replaced by another nucleotide, then it's a DNA alteration. So it is not a deletion. So it is actually the FISH can detect deletion. It can detect amplification, and also it can locate a specific gene to a particular chromosomal loci. So these are the uses of FISH. It cannot detect a DNA alteration. So the answer is RFLP and pyro sequencing. So shortly what I want to say is that whenever you are learning molecular biology technique, it's not possible for you to learn all the techniques. But in general, what we can keep in mind is that we divide the molecular biology techniques into different types, like hybridization technique, sequencing technique, like that, amplification techniques, like that. Then under that, so first thing is that you have to divide into hybridization, amplification and sequencing, and under that you write the name of these molecular biology techniques. For example, amplification, the recombinant DNA technology and DNA cloning, it's an amplification. PCR, it comes under amplification, and you write the different variants of PCR. Then hybridization, blotting technique, FISH, then microvariate technique. These are all hybridization techniques. Then we know that sequencing, DNA sequencing, under that comes the Sanger sequencing, then comes the chemical Max and Gilbert method of sequencing. Then pyrosequencing is also under the DNA sequencing. All these are DNA sequence techniques. So just write down the name of this technique and write the uses of each of this technique. So you make a chart like this, prepare a chart like this, and before the exam you learn just that, because most of the time the technique or how is it being done is not asked, but the use of the techniques are asked. So this is what you have to keep in mind. That is, you write the name and write the uses. Okay. So next is a very highly repeated topic. Various exams this is asked. Thermic effect is highest in. You all know thermic effect is highest in proteins. So what is this thermic effect? Thermic effect means that it is the amount of heat produced by the food during the process of digestion and absorption, uh, which is above the basal metabolic rate. So whatever heat is generated by the processing of the food, like in the digestion, absorption and its metabolism, that is called as the specific dynamic action. It's called the specific dynamic action. It's also called the thermic effect of food, otherwise diet-induced thermogenesis. Okay. So, the heat generated by the process of digestion and absorption and other metabolic activities varies for various macronutrients. Carbohydrate, it is 5%, maximum it can goes to 15%. Okay. Lipids also 5 to 15%. Almost same. But about protein, it is 30%, which means that the amount of heat produced by the digestion, absorption and the metabolism of protein is more than carbohydrate. So, in a mixed diet, where we don't know how much is carbohydrate, lipids and proteins, generally we take it as 10%. So 10% of basic metabolic rate, we count it, or we go like energy, 10% we take it as the specific dynamic action. I hope that's clear. So along, in the thermic effect of food, what I should discuss is that what are the areas that you can expect a question on. So usually the energy content of the food, like the fat, carb and the proteins. What is the calorific value of carbohydrates, fats and proteins, very commonly asked. Then another question that I have seen is the respiratory quotient. So respiratory quotient of carbohydrate, then the fat, etc. is usually asked. So these are the two areas that you have to study extra along with the thermic effect. So that is it. Next we go to the third question. A 12-year-old boy with the type 1 diabetes mellitus presented with unconsciousness. His urine is subjected to a test. The result is shown below. Okay, what is the test done here? So, in a type 1 diabetic patient presenting within a comatose state, what we expect, or what we see, is the first thing that always we should exclude is hypoglycemia, because type 1 diabetic mostly insulin dependent patient, so insulin overdose, mostly what we should expect is hypoglycemia. So first, after ruling out hypoglycemia, then what we should have in our mind is a complication of type 1 diabetes. So what are the complications? The most important complication of a type 1 diabetes mellitus is the DKA, diabetic ketoacidosis. So in diabetic ketoacidosis, the basic test that we can do is urine. In the urine, what we can do is the presence of glucose in urine. So in the presence of glucose in the urine, we can do by a simple semi-quantitative test, and that is Benedict's test. Okay. Then the next one is the ketone bodies. So for the ketone bodies, it's a bedside test that we do is a Roth-Eras test. Similarly, for the urine glucose or the urine reducing substance, we can do a dipsticks also. Similarly, in Roth-Eras test also, instead of doing a test tube, a test inside a test tube and all, what we can do is there are keto sticks. So urine dipsticks for glucose is there, urine dipsticks for ketone bodies also is there available. Okay, so in this picture, we are not seeing any keto sticks or glucose sticks. What we are seeing is a test where a purple ring is seen in the middle of or at the junction of two liquids. So what is it? You all know it. It is Roth-Eras test. So the point here is in the option there is also Benedict's test, but this is not Benedict's test. This is Roth-Eras test. So the answer here is Roth-Eras test, that is answered by ketone bodies. And what is this Fauchet's test? Fauchet's test is a test for bilirubin that we are not expecting in a patient presented with in a comatose state in a type 1 diabetes mellitus. Fauchet's test, if a picture is given, how will you identify? So it's a filter paper test. It's a filter paper test. Okay, that's the first thing that we should be knowing. Okay, inside the filter paper, what we can see is In you can see a white. I'm not able to draw white color here, so I'm just drawing a yellow color here, but actually it's not yellow, it's a white precipitate. So in the middle of this white precipitate, we can see a blue green pigmentation. Okay, so that is, no, again I'm saying this is not yellow, this is white. Okay, so in the midst of a white precipitate, we are seeing a blue green color, that is Faucher's test. Simply you just remember it's a filter paper test. Then Selivanov's test. What is Selivanov's test? Selivanov's test is a test for fructose. Okay, so in Selivanov's test, what we expect is, or what we should get as a positive reaction is a red color. Red color is seen in the test tube. That means that it is Selivanov's test positive. It's a test for fructose. One more test is there for fructose, and that is rapid furfural test. Okay. So these are some of the tests in biochemistry. So important, very important that we should be knowing is the Benedict's test and the Rothera's test. Very frequently asked. So we'll just discuss few tests in biochemistry. That is, the what you have seen is a ring test. So which are the tests that we can expect a ring? One thing that we have already seen is a Rothera's test, where we can see at the junction of two liquids there is a purple ring, and extra what we can see is, you can see crystals of ammonium sulfate in the bottom of the test tube. Ammonium sulfate and a purple ring. So this is the characteristic feature of Rothera's test. Rothera's test. Okay. Okay. Now we go to the next one. Another ring test in biochemistry is aldehyde test. Aldehyde test is for what? It's a test for tryptophan. Indole ring in the tryptophan is the one which is answering an aldehyde test. So here also what we can see is a ring at the junction of two liquids. Again, remember how we can find out it's an aldehyde test. Usually aldehyde test is for tryptophan. Usually whenever we are suspecting, what we can see is the excretion of protein in urine. So usually there will be excretion of protein in urine. Now whenever there is an excretion of protein in urine, what we can see on the surface of this liquid will be froth. So if you can see a froth on the surface, then it's an aldehyde test. And again, what we can keep in our mind is that, so if it's an aldehyde test, usually it will be a history, something related to an amino acid urea, which is related to tryptophan. So that is one thing that we have to keep in mind. So if we are expecting, or if the scenario is pointing towards an amino acid urea, then that ring test should be aldehyde test, because most of the time we may not be able to find out this froth and all in the picture. Okay, the third ring test is Molish test. Molish test. What is Molish test for? This is a general test for all carbohydrates, whether it is polysaccharide, monosaccharide, all carbohydrates can answer Molish test. So how will you find out Molish test? Molish test also, you can see a purple ring at the junction of two liquids. Again, one differentiating feature for Molish test is that, one, the history, the scenario that is given to you will be something related to something which is related to excretion of carbohydrate in the urine. That's the first thing. Second thing is that, I've told you that in Rothera's test there are some crystals here, ammonium sulfate. So while doing Molish test, we are not adding any crystals like ammonium sulfate crystal. So, what we have is a urine and Molish reagent and a concentrated H2SO4. These are the things, reagents that we are adding. So, there is no such things here. So, it is clear. It's a clear bottom with a purple ring. Then it is Molish test plus the scenario. Okay, so these are the main 3-ring test in biochemistry. Okay, so apart from that, let me discuss 3 scenarios which you can get in various questions. The first one is, if in the scenario it's given like Benedict's test is positive, Benedict's test is positive, Rothera's test is positive, then what will be that clinical condition? Then usually it is a diabetic ketoacidosis. Okay. Another scenario, if we are given like it's given and it's given like Benedict's test is negative, but Rothera's test is positive. Then what is it? If this is the case, then it should be starvation ketosis. Starvation ketosis. Okay, so after a strike or hunger strike, a patient is brought, Benedict's test is negative, Rothera's test is positive. So that is starvation ketosis. Apart from that, we also should keep in mind if it's a child presented with hypoglycemia plus ketosis. So a child brought with blood sugar level is nil. Usually, sometimes Benedict's test may not be given. Blood sugar level is very low, but urine ketone bodies are positive. So what is it? This should be a glycogen storage disorder. So the specifically it is a type 1 glycogen storage disorder. It can be 6 also. So it is type 1 because that's the most common glycogen storage disorder. That's what I'm writing here. Von Guericke's disease. So that is hypoglycemia is there, but ketosis. Okay. Now the third scenario is Benedict's test is negative. So there is hypoglycemia. Okay. There is hypoglycemia and there is no ketosis. So what is this? This is usually a defect in fatty acid oxidation. This is fatty acid oxidation defect. For example, medium chain acyl coenzyme dehydrogenase deficiency, where we know that there is decreased fatty acid oxidation. So, there is decreased acetyl coenzyme A and there is decreased ATP production. So, because of decreased ATP production, this cannot, like once the glycogen stores are depleted, there is no ATP to support gluconeogenesis. So, there is no ATP to support gluconeogenesis, and as a result, there is hypoglycemia. Okay. Now, because of decreased fatty acid oxidation, there is decreased acetyl coenzyme A, so there is no ketone body synthesis. Okay. So, if this is the case, then mostly it is fatty acid oxidation defect. One more is the Jamaican vomiting sickness. Then carnitine deficiency. So, all these are possibilities for this scenario. I hope that's clear. So, it's a very, very important area of applied biochemistry. I have just covered most of the topics that can be asked related to this question. So, going to the next question. A patient came with the complaints of muscle weakness. Doctor found that he has cardiomyopathy, diagnosed to him to be a case of Barth syndrome. Where the patient has mitochondrial dysfunction. What is the defect in this? So here the defect is a defect in cardiolipin. So let me discuss a few things about the Barth syndrome and the cardiolipin. So cardiolipin is a phospholipid. phospholipid. And what is it? It is diphosphatidylglycerol. Diphosphatidylglycerol. That means 2 phosphatidic acid and one more glycerol. So why it is called cardiolipin? Because it is first isolated from cardiac muscle. Okay. Then why it is related to a mitochondrial disorder? Because it is most abundant in inner mitochondrial membrane. Okay. So if there is a defect in metabolism, defect in cardiolipin, what will happen? It affects the mitochondria. So it is a mitochondrial dysfunction. So this mitochondrial dysfunction can affect smooth muscle. It can affect the cardiac muscle. Also skeletal muscle. Okay. Now what is this otherwise called as? This Barth syndrome is otherwise called cardio skeletal myopathy. Cardioskeletal myopathy. So in the history what is given is cardiomyopathy. So that is about Barth syndrome. Now what are the other disorders associated with this cardiolipin? So apart from Barth syndrome, it is also associated with aging. It's associated with heart failure. And hypothyroidism. So cardiolipin is associated with all this disorder plus Barth syndrome. Okay. Now a few thing about cardiolipin is that it is the only one antigenic phospholipid. Okay. It's the only one antigenic phospholipid, and this is actually cross react with the antigens of Treponema pallidum. Okay, so what is the problem? It gives false positive test in syphilis. Okay, so that is another point regarding the cardiolipin. Okay, so about all the phospholipid I am saying very shortly. Phospholipids are divided into glycerophospholipid and sphingophospholipid. Okay, glycerophospholipid is made up of glycerol plus 2 fatty acid. Then we call it as diacyl glycerol. 3 fatty acid we call it as triacyl. This is 2 acyl group, so diacyl glycerol plus a phosphate. So diacyl glycerol plus a phosphate plus a base. This base can be a nitrogenous base or a non-nitrogenous base. So this diacyl glycerol plus phosphate we call it as phosphatidic acid. Phosphatidic acid. So phosphatidic acid plus a base, that is called as a glycerophospholipid. Now if this base is choline, then we call it as phosphatidylcholine. What is phosphatidylcholine? That is lecithin. So what is the peculiarity of lecithin? This is the most abundant phospholipid in cell membrane. Okay. Then we also now know that it is the most abundant lung surfactant. lung surfactant. Okay. Now, if the base is ethanolamine, then it is phosphatidyl ethanolamine. Then what is it called as? It is cephalin or kephalin. What is cephalin? Cephalin is a phospholipid present in the cell membrane. Okay. Now, one more option in the question is lysolecithin. Lysolecithin. What is lysolecithin? So, what I have told you here is lecithin, right? So, what we can see is that lecithin has a phosphatidic acid plus a base. Now, phosphatidic acid contains a glycerol plus 2 fatty acid plus phosphate. Okay. So, if one fatty acid is removed from lecithin, then it is called lysolecithin. Okay. So, where is or what is the clinical significance of lysolecithin? So, oxidized lysolecithin is associated with the atherosclerosis. That is the peculiarity, okay, of lysolecithin. And what I have left here is the sphingophospholipid. So, in the sphingophospholipid, just remember there is only one sphingophospholipid, and that is sphingomyelin. Okay, so that is very shortly about the phospholipid. So going back to the question, what we have seen is it's a case of Barth syndrome. It's related to cardiolipin. It's not related to cephalin. Because defect in cephalin is not associated with any of those things. Lecithin, what is the disorder associated with lecithin? It is a respiratory distress syndrome. It is associated with the defect in lung surfactant. So, lecithin. Lysolecithin, I have told you, atherosclerosis. So, this is A is the answer. So, going to the next question, xanthurenic acid is due to deficiency of which of the following vitamin? The answer is pyridoxine, otherwise vitamin B6. So, how is it related? We'll see. So, we know that it is related to tryptophan metabolism. Okay, so we know that in the catabolic pathway of tryptophan is converted to N-formyl kynurenine by the enzyme tryptophan pyrrolase, otherwise tryptophan dioxygenase. Okay, now this is a iron containing metalloporphyrin, this enzyme. So it is a or otherwise we say it's a heme containing enzyme. It's a heme containing enzyme. Now the N-formyl kynurenine is converted to kynurenine. So here the tetrahydrofolic acid will come and it is converted to formyl THFA. And the enzyme here is N-formylase, kynurenine formylase. Okay. Now this kynurenine is converted to 3-hydroxykynurenine by kynurenine hydroxylase. Kynurenine hydroxylase. Okay. This 3-hydroxykynurenine is converted to 3-hydroxy anthranilate and hence it is called kynurenine anthranilate pathway, and thereby this is entering into the ketogenic fate. Tryptophan enter into ketogenic fate, and in between what we should learn here is this enzyme, that is 3-hydroxykynurenine is converted to 3-hydroxyanthranilate by the enzyme kynureninase. Okay, so this is a B6 or a PLP requiring enzyme. So if there is a deficiency of PLP, what will happen? There is decreased formation of 3-hydroxyanthranilate. So if the 3-hydroxyanthranilate is not formed, then what will happen to this 3-hydroxykynurenine? It is converted to xanthurenic acid. Xanthurenic acid or xanthurenate. Okay, so if there is a B6 deficiency, there is excretion of xanthurenic acid. Also remember that is in this step. That is, this 3-hydroxyantranilate is the one which is converted to NAD+. So, if there is a B6 deficiency, what also will happen? There is decreased synthesis of niacin. So, if there is decreased synthesis of niacin, what is it called as? It is called as pellagra. So, pellagra can be caused by B6 deficiency, which is causing a niacin deficiency. Okay, so this is it. Then I just want to cover most of the urinary metabolites that we expect in various vitamin deficiency, which will help you to answer many questions which is related to vitamin deficiency. Okay, so B6 is what is asked here. So in B6 deficiency, which are the urinary metabolites that we are expecting? Already we have seen there is xanthurenic acid. Xanthurenic acid. Then in B6 deficiency, there is oxaluria. Oxaluria, because there is one transamination, glyoxylate, alanine, aminotransferase. Aminotransferase. This is an enzyme which is a transaminase enzyme. Transaminase enzyme require what? B6. So, if there is a deficiency of this enzyme, there is excretion of oxalate in urine. So, oxaluria. The third urinary metabolite that you can expect in B6 deficiency is homocysteine. Why? Because we know that homocysteine is converted to cysteine plus homoserine. This is homoserine by two reaction, and two enzymes in that is, one is cystathionein beta synthase, and one more is that is cystathionein. So what I'm highlighting is cystathionein beta synthase here, because that is a PLP requiring enzyme. So if there is a deficiency of PLP, then homocysteine is accumulated. So these are the three urinary metabolites. Now what about folate deficiency? In folate deficiency, number one is formiminoglutamate acid, that is FIGLU. Because we know that formiminoglutamate acid is converted to glutamic acid, and this comes in histidine metabolism. So, histidine metabolism, this FIGLU is formed. So, for this we need THFA, which is converted to formimino-THFA. Okay. So, if there is a deficiency of B9 or tetrahydrofolic acid, FIGLU is excreted in urine if we are doing a histidine load test. So, if a histidine load is given and FIGLU is estimated in or analyzed in the urine. The next thing that urinary metabolite that we get in B9 is homocysteine. Homocysteine, because we know that the homocysteine being converted to methionine. For that we need THFA, tetrahydrofolic acid, and B12. So, if there is a deficiency of tetrahydrofolic acid, homocysteine is excreted in urine. So, that is about the folic acid, B9. Next is B12. So, if there is a defect in B12, I have told you B12 is also required for this reaction. So, if this reaction is not taking place, one is homocysteine. Then another one is methyl malonate, because B9 is required, B12 is required for a reaction methyl malonyl coenzyme mutase, which convert methyl malonyl coenzyme to succinyl coenzyme. So, if that is not taking place, methyl malonic acid is excreted. So, that is about B12. So, these are the important urinary metabolites that you can learn in various vitamin deficiencies. So, the next question is, in Pompey's disease, the enzyme deficient is. It's a very like a common question. You all know the answer. But most of the time, the problem comes as the name of the enzyme. What we usually know is lysosomal acid maltase. This is the enzyme defect. Yes, this is the enzyme defect, but the same enzyme has one more name, and that is acid alpha lysosomal acid alpha 1,4 glucosidase. Okay, so the same enzyme, and here acid maltase is not given in the option. Like this is alpha 1 glucosidase is asked. This is actually alpha 1,4 glucosidase. So the answer here is 1. What about muscle glycogen phosphorylase defect? It causes what? It causes McCardle's disease. That is type 5 glycogen storage disorder. So what about phosphofructokinase defect? Erythrocyte and muscle phosphofructokinase defect causes Taruv's disease. Taruv's disease. Lactate dehydrogenase defect is not associated with any disorder. Okay. So in Pompey's disease, already we know that it's a lysosomal storage disorder. Which type of glycogen storage disorder? It is a type 2 glycogen storage disorder, and it's coming under muscle glycogen storage disorder with the hypertrophic cardiomyopathy. And usually if a scenario is asked, it will be a severely ill child which is having hypotonia, or it will be given like a floppy infant, macroglossia is a feature, then cardiomegaly. So all these features are if it's asked, then it is more or less it's a provisional diagnosis is Pompey's disease. Enzyme defect is acid maltase, otherwise alpha 1,4 glucosidase. The next question is arrange the steps of karyotyping in order. Even though we don't know anything about karyotyping, from the arrangement from the question itself we can understand what is the steps, because most of the time if you want to do a karyotyping, first what we should do, we should isolate the cell. For example, whatever cell, bone marrow or whatever is the cell that we want to study the DNA, we will isolate that cell. So that is the first thing. The next thing is that karyotyping, then we transport it to the cytogenetic lab. And once it is transported, what they are doing is that they divide the cells into two. We need dividing cells. We don't need the non-dividing cells. So using certain chemicals, we divide or we separate the non-dividing cells and the dividing cells. That's the next procedure. So once we harvest the dividing cells, what we do is that we have to culture the cell. Like we have to promote the division, like the growth of the cell, or in the same like the cell division, various phases of cell division, so that we are incubating, we are culturing the dividing cells by incubating in a appropriate temperature for a few days. That is the next thing. Okay, now then what we have to do, we have to check the chromosomes in the metaphase. So if you are doing usually we do it in metaphase. So if we want to do it in metaphase, what we should do, this is a time where we can get a clear picture of all the chromosomes, where you can see the two sister chromatids joined by the centromere. So that's a metaphase. So we have to arrest it. So to arrest it, what we add is colchicine. Then we study it, stain it, and study. The next procedure is staining. If we arrest at metaphase, we cannot see it. So if you want to see it, we have to stain it. So based on what is the banding technique that we are using, whether it's G banding or R banding or NOR banding. So various techniques are there. The most commonly used is the GMSA stain, where G banding technique, where chromosomes we can see as a dark and light bands we can see. So that is karyotyping. Then what we do is that we arrange we take the chromosome out of the cell and we arrange it in the descending order of its size. Like the largest will be the first chromosome, that is the largest chromosome, we name it as chromosome 1. So like that we arrange all the chromosome 1 to 22, that they are the autosomes, and finally we will put the sex chromosome, that is X or X and Y. Okay, so that is karyotyping. If by common sense itself, we know that these are the steps. So just arrange it, collect the cells. So actually in this question, it's already given in the correct order. Exactly in the question, I don't know in what order it was asked. So this order that I've given is the correct order itself. Collect the cell, okay. Then culture, yeah, dividing and non-dividing cell, culture. In culture media, phytohemagglutinin is used here. Incubated 37 degree for 3 days. then harvest it with Collagen because we want to arrest it, and routine staining with GMs. So this is the order of karyotyping. I hope it is clear. So apart from that, you should also learn the various banding techniques that is also asked in the previous year. So the next question is, which of the following is false about collagen? Phenylalanine is the most abundant amino acid in its structure. No. In collagen, we know that it is glycine XY repeat. So, in a collagen, every third, it's a recurring amino acid, is glycine. So, every third amino acid is glycine, which means that 33% of the total amino acid content in the collagen will be glycine, not phenylalanine. So, that is a false statement. Prolyl and lysyl hydroxylase involved in the post-translational modification. Yes, it's a true statement. Most common fibrous protein. It's the most common abundant fibrous protein in animals and in humans. So it's a true statement. Triple helix structure is important for its function. Yes, triple helix function is a very important structural feature of collagen. So that's also a true statement. So we'll discuss a few things. That is, whenever we are learning about collagen, what are the things that you have to learn? One is the types of collagen. Type 1, where do you find type 1, type 2, type 4, type 7? These are the most important thing, types of collagen, that 1, 2, 4, 7. Okay. Which is the collagen that is found in basement membrane? It is type 4. So that is very important. The second in the collagen, the most important thing is its structure. So in the structure of collagen, very important terms that you should be learning is one which I've already told you, the glycine XY repeat. So this is the primary structure of collagen, where this is the structure, and it is this glycine XY repeat forms a alpha chain, not alpha helix, it is alpha chain. You know, alpha helix is a right-handed helix. This alpha chain is left-handed. Okay, so this glycine XY repeat, which is a sequence of amino acid, which is around 1000 amino acids, it forms a left-handed turn, and that is called alpha chain. Now, the 3 alpha chain, so 3 alpha chain, it is turning in a right-handed direction, and it forms what? Triple helix. So this is another characteristic feature. So in triple helix, what we can see is 3 alpha chains like this. So what we can see is there are certain constricted areas here. So what is the importance of glycine? The glycine is occupying all the constricted areas. So it's the smallest amino acid. Only glycine can occupy those constricted areas. So glycine is very important for the alpha chain and for the formation of triple helix. So this is triple helix. Now once the triple helix, so we have lots of triple helix. It forms a particular lateral association. Like it is called a quarter staggered arrangement. Okay, quarter staggered arrangement. So these are the structural features. In the quarter staggered arrangement, we can see that this is one triple helix, another triple helix. The second layer of triple helix will be 1/4 distance away. The third layer of triple helix will be again 1/4 distance away. Like that, it's a quarter staggered arrangement. And in the quarter staggered arrangement, there is one more feature, that is, there is interchain and intrachain covalent crosslinks. So it is also important for the tensile strength of collagen. Okay, so these are the structural features. Next very important thing is the synthesis of collagen. So in the synthesis of collagen, what we should understand is that there are two phases. Intracellular and extracellular. Okay. In the intracellular, which is the cell involved rough endoplasmic reticulum of fibroblast. This is the site where the intracellular events are happening. So in the intracellular events, the most important changes are the glycosylation. Then glycosylation takes place in hydroxylysine residue. This is a type 3 O glycosidic linkage. Then another important feature or post-translational modification that was in the question, hydroxylation. Hydroxylation of proline and lysine residue. By which enzyme? Prolyl and lysyl hydroxylase. Okay, so for this, which is the vitamin important for this? Vitamin C and alpha ketoglutarate. Okay, then then these are the important things, and finally this is forming the triple helix, that is also seen as an intracellular event. Okay, now the triple helix, which is called as a procollagen, is extruded out, or it is given out of, or it's going out of the rough endoplasmic reticulum, out of the cell into the extracellular matrix. So in the extracellular matrix, the major events are what I have told you is the quarter staggered arrangement. Okay, so this is the quarter staggered arrangement. So quarter staggered arrangement, very important feature is the covalent crosslink. So what is the post-translational modification important for this covalent crosslink? It is oxidative deamination of lysyl and hydroxylysyl. Oxidative deamination of lysyl and hydroxylysyl. So for that, which is the enzyme? Lysyl oxidase. Okay, so for this lysyl oxidase, which is the very important cofactor? Copper. So copper is also important for the tensile strength of collagen. So copper deficiency and vitamin C deficiency can cause collagen defect. The next thing that we should learn in the collagen is the collagen disorders. So very important is the osteogenesis imperfecta, which is associated with the type 1. Then Alport's, very frequently asked. Alport syndrome associated with the type 4. Then epidermolysis bullosa, which is dystrophic. This is associated with the type 7 collagen. Okay, then Ehlers-Danlos syndrome. There are various types of Ehlers-Danlos syndrome where it will be difficult for us to remember all the Ehlers-Danlos syndrome because almost every collagen is associated with an EDS. So most of the time these are the defects that is asked, and apart from that, Menkes disease, which is a defect in lysyl oxidase. Which is caused due to a copper defect. So these are the major collagen disorders. And scurvy, how scurvy is affecting? Scurvy, which enzyme is affected? It is the lysyl hydroxylase, not lysyl oxidase. Scurvy also causes collagen defect, and that is lysyl hydroxylase. Okay, so these are the major areas that we have to learn in collagen. So going to the next question, ketone bodies are not utilized by liver because it lacks. So it lacks an enzyme thiophorase, and that is the answer. What about the other enzymes given here? Thiokinase. Thiokinase is an enzyme involved in fatty acid synthesis, that is acyl coenzyme A synthetase is the one which is otherwise called thiokinase. So it is not related to ketone body utilization. Then thiotransferase is not an enzyme involved in ketone body synthesis or utilization, so that's also not the answer. Then thiolase. Thiolase is involved in ketone body utilization, but this is not an enzyme that is absent in the liver, because we know that thiolase is also involved in fatty acid oxidation. Fatty acid oxidation is a pathway that is taking place in the liver also. So it is not an enzyme. It is an enzyme involved in ketone body utilization, but it is not an enzyme which is absent in the liver. So hence that is also not the answer. So here the answer is thiophorase. So we will just see about the ketone body synthesis and utilization. So ketone body synthesis is a pathway that is taking place exclusively in liver mitochondria. Very important, liver mitochondria. Okay, ketone body synthesis happens as a continuation of fatty acid oxidation if the body demands so. Okay, so as a continuation, we know this is the fatty acid oxidation, and we know that acetyl coenzyme, the two carbon acetyl coenzyme is released one after the other, and finally it will stop at the point called acetoacetyl coenzyme. This is the starting substrate for ketone body synthesis, the starting point of ketone body synthesis, where we can see the 2 carbon acyl coenzyme, which is called acetoacetyl coenzyme, combines with the acetyl coenzyme to form a 6 carbon compound, and that is called HMG coenzyme. So for this, the enzyme is HMG coenzyme synthase. Then it is undergoing the next step, which is called HMG coenzyme lyase, where the primary ketone body called as acetoacetate is produced. Okay. Now this acetoacetate is converted to beta hydroxybutyrate and also to acetone. And acetone and beta hydroxybutyrate are the secondary ketone bodies. Here the enzyme, this is a non-enzymatic process, or it is a spontaneous decarboxylation, whereas acetoacetate to beta hydroxybutyrate is an enzymatic process where beta hydroxybutyrate dehydrogenase is the enzyme there. Okay, so these are the secondary ketone body, and in this ketone body synthesis, the rate limiting step is HMG coenzyme synthase. Okay, now once the ketone bodies are synthesized, it is never utilized inside the liver, because the utilizing enzyme is not there in the liver, especially the one enzyme, that is thiophorase. So where is it coming? So what we have seen is these are the ketone bodies. Acetone is a different ketone body which is not like excreted through the urine. Then how is it excreted? Acetone is a ketone body that is excreted through lungs. So it is expired because it's a volatile ketone body, acetone. Now, whereas the other one, like the acetoacetate and the beta hydroxybutyrate, are the ketone bodies which are excreted through the kidneys. Okay, so how is it utilized? It is not utilized by the liver, but by the extrahepatic tissues only, and not by the liver, and also one more organ. What is that organ? RBC. So apart from that, all the other organs can utilize ketone bodies. So beta hydroxybutyrate and acetoacetate, we have seen that how is it produced. The beta hydroxybutyrate, whatever is produced, is converted back to acetoacetate. Now, acetoacetate means that there are this is a 4 carbon compound. So, there are 2 potential acetyl coenzyme. So, finally what happens is this 2 acetoacetate is converted to 2 acetyl coenzyme. So, for that we need a few enzymes, that is acetoacetate has to be converted to acetoacetyl coenzyme. For this, we need an enzyme called succinyl coenzyme acetoacetate coenzyme transferase, or this is otherwise called as thiophorase. So here acetoacetate to this a coenzyme is transferred. This enzyme is thiophorase. This enzyme is lacking in the liver. So, liver cannot utilize ketone bodies like the acetoacetate or the beta hydroxybutyrate, because both has to come to the stage called as acetoacetate. Now, during this step, what we can see is that a coenzyme is transferred from succinyl coenzyme. Okay. Now, succinyl coenzyme to succinate, we already know it's a step of TCA cycle, where we can see that that was a step normally in TCA cycle, it is done by an enzyme succinate thiokinase, where we can see that it is a substrate level phosphorylation where GTP bar ATP is produced. So if there is ketone body utilization, that step will not take place. Instead of that, thiophorase is the one which convert succinyl coenzyme A to succinate, and there is no production of ATP here. Okay, now this acetoacetyl coenzyme is converted to by the enzyme thiolase. So that's what I said. Thiolase is coming in fatty acid oxidation as well as in ketone body synthesis, ketone body utilization. But remember, this thiolase is present in the liver. Okay, now this acetyl coenzyme is again getting into the TCA cycle. Okay, so what we have seen is that from acetoacetate we will get 2-acetyl coenzyme. 2-acetyl coenzyme, we know that 2-acetyl coenzyme, if it enter into TCA cycle, normally into TCA cycle, how many ATPs we will get? We should get 20 ATPs, right? 10 into 2, 20 ATPs. But here what we have seen is that from 2-acetyl coenzyme we are getting 20 ATPs, but one acetyl, like the TCA cycle that is seen here, one step of TCA cycle is altered or it's replaced by another step called thiophorase for ketone body utilization, which means that one ATP is not generated here because that substrate level phosphorylation is not taking place. So what we expect is 20, but we are not getting the succinate thiokinase step, and hence we have to minus 1 ATP from there. So net we will get only 19 ATPs from what? Acetoacetate. Okay. So what I've told you is acetoacetate is 2 potential acetyl coenzyme. 2 potential acetyl coenzyme means 2 TCA cycle, means 20 ATPs. But we have seen that in one step succinate thiokinase is replaced by thiophorase, which will not generate an ATP, and hence we have to subtract 1 ATP. So how many ATPs we will get from acetoacetate? We will get 19 ATPs. Okay, so from beta hydroxybutyrate, how much we'll get? Like from beta hydroxybutyrate, we'll get 21.5. Why? Because beta hydroxybutyrate on conversion to acetoacetate, 1 NADH is produced. 1 NADH, we know that it is 2.5 ATPs. So from acetoacetate we are getting 19. So from that we are adding one more 2.5. So 19 plus 2.5 is 21.5. Okay. So from one acetoacetate we will get 19 ATPs, and from one beta hydroxybutyrate we will get 21.5 ATP. So that is about ketone body utilization, and this is how ketone bodies can produce or generate ATP. Okay. So that is about the ketone body synthesis as well as utilization. So going to the final question, a template DNA strand is so and so. What is the resulting mRNA? So if you want to answer such questions, so two things that we have to keep in mind. Number one is it is a template strand. So whatever RNA is the complementary to the template strand. So we should be following the Watson-Crick base pairing rule and find out the complementary sequence. Number two is that the reading of the template strand is taking place in the 3' to 5' direction, and synthesis of RNA is taking place in 5' to 3' direction, which means that we have to make this 3' the 5' end. That's the first thing that we have to keep in mind. Okay, so first find out the complementary, and we are making this 5', and now we are finding the complementary sequence where we can see that the C is pairing with G, T with A, U, C, G, A, U, A, U, U, G, A, U and G, and make this the 3' end. Okay. Now in the question 5' to 3' and 3' to 5' is given. So here we should always see which is the direction. Like GUAAA, like that, if it is given in the option, it is not the answer because GUAA is from 3' to 5' direction. So the direction of the newly synthesized RNA is also important here. So first option is GA. So we can make this 5'. From here we have to read G A, U, C, G, A, so and so. We can see that the option A is the correct RNA sequence. I know that without having any paper or rough place to work out during your exam, it's very difficult to find out the answer of such question. If I'm answering this question also several times, I myself will make mistake here. So this question is easy, but the problem is we have to take some time to answer such question. Don't be in a hurry to answer such questions, because you have to see the direction, we have to see the sequence. If it's a 3 or 4 sequence, then it's easy, but more than 10 bases are given, and if it is like this what is given here, then it is difficult. So you have to accept this fact, and when you answer this question, it's easy. If you know what is it, then we should take some time to answer such questions. So before concluding, what I want to say is change your mindset. It's a game changer. So from in the mind, if we have any negative thoughts, always just extrude those negative thoughts and fill your mind with positivity. And a positive man, like a person with positive mind, is actually a very powerful man, which who can do so many things. So nothing is impossible if you have a positive mind. So always be positive and have a positive attitude to all the failures and all the negative things in your life. Thank you so much.